Good day, everyone, and welcome to another laboratory session in clinical bacteriology. Today, we will discuss the importance of a bacterial smear preparation in starting a staining process before doing microscopic examination of bacterial cells. Now, in definition, bacterial smear po is a thin layer of bacteria placed on a glass slide and fixed by heat partly to ensure that they remain attached to the glass for staining procedure. Now, preparing the smear requires attention to a number of details that help prevent contamination of the culture and ensuring safety to the preparer. Now, each step of the procedure has an important reason, and each should be followed carefully as a prerequisite to successful work. Let's take a look at the different types and considerations in preparing a bacterial smear. Now, please take note that bacterial smear can be made from either pure cultures of bacteria or directly from specimen. Now, when it comes to preparation of bacterial smear, using a specimen, we do this glass to ensure the quality of the specimen we receive. Now, when we do naman po bacterial smear preparation using pure cultures, now, usually po it can be prepared from a solid medium or a liquid medium. So when we say solid medium po, um, our source po would be the colonies growing on the plated media. Now, when we say liquid medium naman po, these are bacterial cells growing on a broth medium. Now, what are the different considerations when you are doing your bacterial smear? So first consideration is that the bacteria po should be evenly and lightly dispersed in a glass slide. And then also, the bacteria should be firmly attached to the slide so that they are not washed out during staining procedure. And usually po, we firmly attach the bacteria to the slide by allowing it to dry, no, and then using heat as a source of fixative. So we heat the slide to um, completely dry the whole smear. Now please take note that before drying your smear or fixing your smear using a heat source, you first allow it to dry. That is because bacteria will boil, and then the cell morphology will be lost. Now, be sure also to label the far edge of the slide. Use proper labeling. No, so the two main purpose of the proper labeling is to know which side of the smear is to be stained, and also to um to properly label and to know that the smear would be on a single patient lang. And then lastly, no, allow your slide to air dry before you heat set it. Now, the materials we need to start your bacterial smear preparation are the following. So first would be the pure bacterial culture, solid or liquid type, or specimen. So for bacterial source, no, we usually use pure cultures. When we say pure cultures, these are bacteria no, that a family of bacteria growing together. So it is a single type of bacteria growing together. Now, next naman po would be clean glass slides. Usually, the prepared one would be the frosted one, no, for proper labeling. Also, a pencil for labeling. So, do not use marker or ink pen, no, for labeling the frosted side of the glass slide, no. This is to prevent po washing out of the label, no, by the chemical reagents po that we will employ on the staining procedure. Now, next material would be the inoculating wire loops and needles. These are manipulating devices for you to transfer the bacteria coming from the source and then onto the glass slide. Now, next naman po would be your alcohol lamps. Now, your alcohol lamps po are the direct heat source for proper fixation of the bacterial smear po onto glass slide. And lastly, sterile water in the form of normal saline solution or this sterile distilled water po. This last material po is used po for the emulsification of solid cultures po or solid colony coming from a plated media. Okay, so let's now move on to the step-by-step procedure in doing a well-prepared bacterial smear. Now, first things first, you always remember, class, to follow aseptic techniques no before or within no doing this technique. So when we say aseptic technique class, you do not allow any form of contamination coming from the environment going into your preparation. So every step of the procedure po, you need to um, be aware no, that you need to always sterilize no, your equipments and lessen the contamination coming from the environment. So the first things first is to disinfect your area properly and then prepare the materials to be used. Secondly, now you get a clean glass slide and then um, you do a septic technique no, by passing the slide po to a flame until no moist appears on the slide. Now, the aseptic technique of placing your clean glass slide on a flame also helps po to eliminate po the excess oils or grease in the surface of your clean glass slide. Now, on the third step naman po is labeling. So, you label the slide appropriately using pencil to label the slide. So, again, do not use pencil markers or ballpoints. So, this could be washed out on the staining phase. Now, on the fourth step is you aseptically prepare your inoculating wire loops and needles no, by putting it on a flame. So the wire should be red hot to ensure the sterility of the wire loops and needles. And then these wire loops and needles is used separately based on the source of their bacteria. For solid cultures class, you use needles to get colonies coming from the plate and then transferring it to the glass slide. You use loops naman po when your source of bacteria po is in the form of broth or liquid type media. Then on the fourth step, you allow to cool your inoculating wire loops and needles no, to ensure po that you will not kill no, the bacterial cells before transferring it to the slide. Now on the fifth step, you prepare now your sterile glass slide. Now, for solid cultures po, the sixth step po would be placing a drop of sterile water to the slide. Again, sterile water po would be your emulsifier in this step. So, place a drop of your sterile water to the glass slide and then get solid culture po using wire needles no, and open it aseptically. Now, get the solid culture and open it aseptically by flame flashing the rim of the plate or tube. Now, using an inoculating needle glass, get a small 1 to 2 colonies of the bacterial isolate and mix it with the drop of sterile water until homogenized. Now, how about if your source po would be liquid cultures or broth cultures? Now, the sixth step naman po if your source would be a liquid culture is that you get the liquid culture po and open it aseptically by flash framing the rim of the tube. Now, and on the seventh step, this time you use an inoculating loop and then get a loop full of the broth and place it directly on the sterile glass slide. So, as you can see here, no, compared to the solid cultures, in your liquid cultures, in preparation of your liquid cultures, you do not use emulsifier or your sterile water po because the broth po serves as the homogenizing media already. Now, on the eighth step po, now on both solid and liquid cultures, the next thing to do po is you spread it evenly to achieve a thin film. Now, sterilize the inoculating wires by putting it on a flame. And then on the ninth step, now you allow it to air dry completely, approximately 3 to 5 minutes. So it should be air dried completely now before heat fixing. So because if you heat fix already po, the bacteria will boil, no, and the cell morphology will be lost, and also to avoid splashing. And then when the cell morphology po um will be lost, no, or if the cell morphology is lost na, it will affect po the staining procedure, no? And then on the 10th step, no, you heat fix the smears by quickly passing the slide through the flame. Now the heat should contact the underside of the slide, no? And then heat fixing po, please remember, no, the functions of heat fixing are the following. So it kills any bacteria that may be still alive, and it also facilitates stain penetration and fixes the cells to the slide so that they do not wash off when stained. Now, and then on the last step, now you proceed now to the staining process. Now, on this slide lang po shows you the properly done smears, no, that has a proper thickness and proper sizing. So please take note, class, no, the size of the smears may vary depending on the source, no, and then depending on the consistency and physical appearance of your bacterial source or your clinical specimen. So please take note, class, if your clinical specimen po is physically it is viscous. So for you to avoid thick smears or producing thick smears coming from a viscous specimen, you always remember, class, that if your specimen is viscous, you um you um you create po a largely smear, or you create your smear as large as possible. This is to allow po for even spreading of the viscous fluid po in your slide. But when your clinical specimen naman po is not viscous, or it is liquidy, or liquid type, like for example, you are processing urine or CSF or any body fluids that is not visibly viscous, you create your smear po smaller as to allow the diluted specimen to be concentrated naman. Now, what if your specimen naman po is turbid, no? You see your specimen as turbid. So if your specimen po is turbid, um in the sense, no, for you to evenly disperse po the bacteria and not to concentrate it, you create a larger smear if your specimen po, or your liquid type specimen po is turbid or opaque already. But if your liquid naman po is not opaque, or it is translucent or transparent, so you allow your smear po in a smaller size. That is to concentrate naman po the diluted specimen. Now, this slide po shows you easily the basic procedures of bacterial smear preparation using solid medium and liquid medium. So as you can see here po, for solid medium, you use inoculating needle. This is not to allow po excess, no, amounts of bacteria to be placed in your preparation. So as you increase kasi the amounts of bacteria that you place, you also increase the chances of producing thick smear. Okay? Now, for liquid medium naman, you use loops, no, to get an ample amount of liquid medium. So, you spread it thinly, no, and then air dry, and then heat fix completely. And in the slide po, shows you the procedures, no, if you are doing a liquid type preparation of bacterial smear. Okay, so let us now see how simple staining is done, no, properly, and what is the purpose of simple staining. Now, the sole purpose po of simple staining is to give a color contrast to the colorless bacteria by using a positively charged dyes, thereby allowing it to be perceived visually by the eye. Now, all types of bacteria appear as color of that stain when viewed under the microscope. So, usually, no, we view again bacteria to determine a bacterial species morphology and arrangement, no? But they do not give any additional information. So simple staining doesn't differentiate, no, groups of bacteria. It just allows po allows observation of the morphology, the basic morphology and arrangement of a bacteria. Now there are two types of simple staining. So namely your positive staining and negative staining. Now, in the simple positive staining, no, you directly stain bacterial cells in its cell wall, and then usually the bacteria appears dark, no, on a bright background. Now, the commonly used basic stains are the following. These are your methylene blue, crystal violet, malachite green, basic fuchsine, carbylfuchsine, and safranin. And these basic stains po are usually cationic dye. No? When we say cationic dye, it is positively charged. Now, in simple negative staining po, in contrast, no, these are stains po that these are procedure, no, usually stains the background instead of the object. So the object appears or the bacteria appears bright or colorless against a dark background. And the commonly used negative stains po are the following. So these are your India ink and Nigrazine stain. Now, the materials for doing a simple staining are the following. So first is your bacterial smear. no, it is done, which is done properly, a timer, your stains, no, usually po a one type of stain, staining rack, water source for washing step. So it is either a water source coming from faucet or wash bottle. Now the prepared one po for washing step usually is wash bottle. And then lastly is blotting papers. Now let's take a look at the step-by-step process of a simple staining. So the first step again is disinfect your area and prepare materials to be used. Secondly, get the prepared slide with the bacterial smear and check the correct side with the bacterial film and place on a secured staining rack. Now the third step would be um, application of the stain. Okay, so you flood the bacterial film with the stain, ensure that the whole smear pot is covered, and then you allow the stain to stay on the smear for about 30 seconds to 1 minute, then depending on the protocols. Now, the fifth step po is to wash off the excess stain by gently passing the slide to a slowly running water. Now, alternatively, you can use wash bottles in this step until the excess stain runs off. Then after doing so, you place the your slide no on a blotting paper and gently dry the slide. Do not rub the slide as it can as it can remove no the already stained smear in your slide. Now on the seventh step, you proceed now to microscopy. Now, lastly, on this slide po, shows you a simpler um, procedure no, of your simple stain. So you begin again with the heat fix emulsion, you cover the smear with stain, and then you wash off the excess stain, and then you dry your slide using a bulbous paper. Okay, that's all for this discussion. I hope you gather information for this topic. Thank you and good day.